It is a pleasure to introduce Dr. Thomas, with whom I've worked in the past. He is a gentleman and a scholar. He's a professor of medicine and a professor of microbiology and immunology and infectious diseases, and a physician scientist from the SUNY Upstate Medical University. He is the director of the Institute of Global Health and Translational Science and the interim chair of microbiology and immunology. Dr. Thomas has specialized in the study of viruses and vaccine development, and he's currently the global coordinating principal investigator for the Pfizer phase 1-2-3 COVID vaccine trial. So with that, Dr. Thomas, please take it away.
Thank you very much, Dr. Heisig. I'm going to come on my camera now just so I can look everyone in the eye and say thank you very much to the New York chapter of the ACP for having me, and thank you everyone for joining, and thank you very much for everything that you do every day. I'm now going to share, and because I'm battling my children for bandwidth here, I'm going to stop my video. Okay, Amanda, can you see my slides?
I sure can.
Okay, perfect. So let's get right into it. As Dr. Heisig mentioned, I'm going to kind of give hopefully a state of the art and sort of a roundup of COVID-19 vaccines and vaccination. These are my disclosures related to COVID vaccine development, in addition to being a principal investigator with Pfizer and BioNTech. Upstate, and my colleagues and I work with a number of other developers, and I do some consulting and sit on safety boards. This is a general overview, so I'm gonna try and touch a lot of different topics that relate to COVID vaccine development in general, and the SARS-CoV-2 virus. Some areas that I think are more timely, I'm gonna dig a little bit deeper into those topics. But first, I think we need to always kind of refresh our memories about where we are with the epidemiology. And this all started, which is really unbelievable to think that this has been going on for almost 2 years now. And I first saw it, as many of you may have, with this sort of ProMed post at the end of December in 2019, where there was a cluster of cases of pneumonia that they could not make a microbiologic diagnosis, and the numbers kept rising. This was in Hubei, China, and this was kind of the shot that heard around the world and started this pandemic. And where are we now? As of the WHO's reporting yesterday, we're at over a quarter of a billion confirmed cases, which we all know is an underestimation. Obviously, there are many more people infected who did not get diagnosed, either because they didn't have access to diagnostics or because they were asymptomatic or minimally symptomatic and never presented to medical care. And unfortunately, we've had over 5 million deaths globally, which again is also an underestimation. The Americas and Europe have made up the vast majority of cases, with Southeast Asia coming in third behind the two of them. Overall, the global trend is one of a gradual decline in activity, but of course there are hotspots. As it relates to the United States, as of this morning anyway, we again are also seeing a gradual decline. It looks like we're on the backside of what has become our Delta surge, and the second largest surge we've had throughout this pandemic. We're averaging about 70,000 cases a day, about 50,000 people hospitalized, and over 1,000 deaths a day, which fortunately all those numbers represent, looking at a 14-day average, look at anywhere from 10 to 20% declines. Again, we've had almost 750,000 people in the United States that have died since the virus first hit our shores. And if you look over at the right, what the last 90 days looks like, again, it's a very slow but gradual decline. Hopefully it continues to decline and we don't just see a plateau of where we are. And the reason why we're not making faster declines is because we still have many hotspots in the country. This is a heat map with the darkest red representing over 250 average daily cases per 100,000 people. And you can see where some of the hotspots are in Montana, North Dakota, Wyoming, Colorado, and Utah. So although lots of the country is seeing improvement, there are still lots of hotspots, which as an as a nation keeps us at that very gradual decline. When we look at New York, this was not, Delta did not result in our largest peak. It's probably the third largest peak that we've seen. And again, fortunately, we're tracking with what's happening nationally, and we're seeing declines anywhere from 5 to 12% declines in terms of deaths, hospitalizations, and the number of cases we're seeing on average. And again, these are 14-day averages. The last 90 days, again, the overall is a decline, but it looks like we're leveling out a little bit. And just like the nation, we have hotspots even within our own counties. So just northwest of Albany and out in the western part of the state. At one time people thought that obviously COVID was, not a big deal for kids, but it actually is a big deal for kids. And if you look at this is from the American Academy of Pediatrics, and if you look at the right side of this data with along with Delta and along with unvaccinated adults getting infected, lots of kids got infected and continue to get infected. And so we're looking at children making up about 20 to 25% of all the cases that are counted each week in the United States, with the light blue representing adult cases and the dark blue representing cases in children. So less than 20 or less than 18, depending upon the state that is reporting. And unfortunately, as of this morning anyway, the CDC had reported over 650 deaths. Males are represented on the left and females on the right. The light blue represents children 0 to 4 years of age, and the dark blue represents children 5 to 18 years of age. And if you take the 2 years of COVID and compare that to 2 years of 2 seasons of influenza, COVID has killed at least 2, probably 3 to 4 times as many children as influenza does. So this is clearly a problem for children. Okay, so I want to talk a little bit about the virus because it's important just to know a little virology because it helps to understand the vaccinology component. So this is an artist's rendition of the virus. It's a sphere. On the inside of the sphere is the RNA, right? The genetic material. It's wrapped around this by an envelope, and there's some proteins that exist on the surface of the envelope, M protein, E protein. But the really important protein is this spike protein, which is represented by the pink projections that are coming off of the virus. And if you look on the right, the spike protein, it's actually a trimer. So it's 3 different proteins that are put together, and they're made up of an S1 subunit and an S2 subunit. There's a stalk, and then it's covered in these sugar molecules or these glycans, which kind of have an immunoevasion function. But it's not a stiff structure. It has with this particular author called the hip, knee, and the ankle, which allows it to move and be dynamic. And it is this spike protein which is so important for infecting human cells, because what happens is we have on our cells these ACE2 receptors. And the spike protein recognizes these ACE2 receptors, and it's like a key going into a lock. And once that key recognizes the lock and the connection is made, there's another component of the receptor that modifies that spike protein and then changes it conformationally so that it can then anchor, the virus can anchor to the cell that it wants to infect. There's movement, and the cell will join to the virus, or the virus joins to the cell. And once that happens, then the genetic material inside of the virus gets into the cell, uses the cell's own machinery, makes baby viruses, which then go out and infect lots of other cells. Okay, so spike protein, remember that. So I just want to talk a little bit about vaccine development in general, because I'm not, I never want to assume that people kind of know the process. I'm a vaccine developer, so I've been doing it for a while, but I think it's been the source of a lot of maybe miscommunication or misinformation, and sometimes not setting appropriate expectations. So going from concept to vaccine, there are recognized phases of vaccine development, and I call the 3 on the left as vaccines and the 3 on the right as vaccination, right? And, this is not my quote, but I like it. Vaccines are not what saves lives, right? It's vaccination which saves lives. And you need both of those components to make a public health impact. So going from concept to vaccine on the vaccine side, so the idea of exploration, and so this is where vaccine developers work with immunologists and pathologists and clinicians and epidemiologists to understand what is the disease, what causes the disease, how do people get infected, what kind of immune response do they develop, who lives, who dies, what are the pathologic or immunopathologic mechanisms. And we use that information to start selecting targets for our vaccine and start making prototypes in the laboratory. And then we move on to preclinical testing, which is typically, test tubes in the wet lab and pipetting and things of that nature, cell cultures. And then we move on with these prototypes to studies in small animals. So it could be hamsters, rabbits, mice, et cetera. And then if it's appropriate, going into larger animals like non-human primates. And this is where you demonstrate your first proof of concept that the prototype is safe and may actually work and have clinical benefit in people. Now, between preclinical and clinical, there's a big regulatory component and a manufacturing component. You can't take laboratory grade vaccine prototypes and put them into people. You have to go through a biomanufacturing process, which is highly regulated by the FDA. But once the FDA gives you the go-ahead, then this is when we go into clinical development. And this is standard 3 phases of testing. Phase 1, which is small numbers of people focused on safety. Could be up to 100 people. Phase 2, which is expanded, it could be hundreds of people. And it's not just focused on safety, it's also focused on is the immune response that we think will protect someone or attenuate a disease process, are we generating an immune response in people? And if you get through those 2 phases, then you go to phase 3, which is the phase where you want to demonstrate not just safety in much larger numbers of people, but also that the vaccine does what it's supposed to do and demonstrate clinical benefit, whatever is defined as clinical benefit. And at the end, if through those phases and through the animal studies, you can demonstrate that your vaccine and the FDA says your vaccine is safe and efficacious, this is when then you get into the much larger scaling production from these small clinical lots that support your phase 1 and phase 2 studies into the big, huge lots. It's one thing to produce something at 2,000, 3,000 doses. It's another thing to produce it at 100 million doses. And once you get a regulatory approval and your vaccine can be sold and it's on a shelf and providers can pull it off the shelf and give it to somebody, then that relationship that you have with the regulatory authorities is now a lifelong relationship, and you are subject to multiple audits and reviews and inspections of your manufacturing facilities, et cetera. And this is the part that supports vaccination, which again, vaccination is what saves lives. So this is the process. This is normally how it goes. And if you look at it above the red line, this is what some people are kind of upset about, is that they say, well, wait, normally that process you just showed me normally takes up to 10 years. It costs about $1 billion. It's a very iterative process. It goes from one phase to another, and there's checkpoints and everything else. How is it possible that you could have gone from the sequence of a virus to an emergency use authorization meeting in 11 months? It doesn't make any sense. You must have cut corners, and it must have, and it's jeopardizing, our safety. But really, if you dig into it, that's actually not the case. So first and foremost, that traditional paradigm at the top, we in the vaccine business have been speaking for a long time about how that it does not have to be that way. The primary reason it is that way is risk mitigation and financial risk mitigation. But it really never had to be that way to begin with. That didn't need to be the baseline to compare it to. But if we just look at what happened with COVID, which is represented here by a much shorter line, which we've all observed and watched, first of all, the technology had a head start. So messenger RNA vaccines, adenovirus vectored vaccines, people have been working on these platforms for 30 years. They've been doing clinical trials in humans with other diseases, so Zika, HIV, influenza, et cetera, for the last 10 years or so. And so thousands of people have already received versions of these vaccines. So the technology had a head start. The second is that there were parallel processes because people were willing to accept financial risk. So, typically you would not go into commercial scale up until you were very certain that the vaccine was safe and very certain that the vaccine was going to have a clinical benefit. In this case, because of circumstances, and because of the fact that through Operation Warp Speed, close to probably will end up being $13 to $15 billion was provided to pharmaceutical companies, they could manufacture at commercial scale at risk. So if the vaccine was proven to not be safe or not be efficacious, they would throw it out. But if it was proven to be safe and efficacious, then it would be available for immediate deployment. The third part or the fourth part is that the very engaged regulators. And so when you embark on this process, you're in constant contact with FDA, and you're constantly asking them, what's my sample size need to be? And these are my endpoints. And do you agree with those endpoints? And, do you agree with our assumptions and these sorts of things? And of course, regulators, because of the pandemic, were very, very engaged. And unfortunately, but fortunate for vaccine development, there was so much COVID that it was very, very easy to collect cases and quickly demonstrate a difference between vaccine and a vaccine and placebo. And people were very interested in volunteering. We at Upstate were one of the COVID trial sites for Pfizer and BioNTech. And we received in the literally 2 or 3 days after we announced that we were a trial site and we were taking volunteers, we had more than 2,000 people that had called, and we ended up enrolling about 400. So that's how we were kind of able to set a new standard in terms of speed of development. And I can tell you, having been in conversations now with vaccine development efforts not related to COVID, there is a new standard and there is a new expectation for the time that it will take to develop vaccine products. So for me, and I think this is one of the areas that we probably did not do the best job of managing expectations or communicating scientific or health information effectively, is, for me, the goals of developing a COVID vaccine was to reduce the occurrence of severe and fatal COVID disease, right? So prevent severe disease, prevent hospitalization, and prevent death, right? That's all encompassed in human suffering. By doing that, we can achieve the second goal, which is to protect the healthcare system so that these surges don't overwhelm primary care offices, don't overwhelm emergency departments, overwhelm hospitals, overwhelm staff. And as a result, then there are secondary and tertiary effects of COVID. And I'm sure everyone on the phone here has been impacted by our inability to do this at times. But of course, there's some additional goals, right? We would love to make a vaccine that prevents infection. We would love to make a vaccine that can actually reduce a transmission between people, because we would like to get our society back. But really, the two in red are what I see as the primary goals. And there are lots and lots of groups that got into and remain in the COVID vaccine development business, if you will. I was involved in making vaccines for Ebola, MERS-CoV, and Zika, and lots of people show up, but they quickly kind of depart. There's been a lot of staying power with the COVID vaccine development initiatives for obvious reasons. But just so you can see, now that you all know the phases of study, you can see there are dozens of vaccines that are in clinical development right now, a number that have been authorized or have received full approval. And there are, every day there's a ticker tape of new happenings in the COVID vaccine world. So it's not just some of the big pharma companies we're always seeing. It's multiple different groups, governments and militaries and academic groups and biotechs. But despite the fact that there are dozens and dozens of different types of vaccines, there really is one common approach, and this gets back to that selection of targets. And remember, I mentioned that you need that spike protein so that the virus can fuse to the cell that it wants to infect. So it stands to reason that if you can block that interaction from happening, so if you can target those epitopes that are on that spike protein, and you can get neutralizing antibodies attaching to those epitopes, you can prevent that interaction from occurring, and you can reduce viruses from infecting cells, from making new viruses, and maybe you can attenuate someone's viremia, and therefore attenuate their disease and maybe even prevent them from being able to infect somebody else. So this is the common target. So how does it work? So these are RNA vaccines, and I'm just going to focus on kind of the United States. I'm not going to get into all the other vaccines that are out there. I'm just going to focus on what's available to us. So once the genome was understood for SARS-CoV-2, they said, well, let's take the section of that genome that codes for the spike protein. We'll take that code. We'll package it in this lipid nanoparticle, and that will be our vaccine. And we'll deliver this lipid nanoparticle with the RNA code inside of it into the person. And once we do that, so once that goes into the arm and into the muscle and into the other cells, the lipid nanoparticle enters the cell. It dumps the mRNA, gets released. The cell says, I understand what this code is telling me. It's telling me to make spike protein. It makes spike protein. It gets expressed on the cell, these antigen presenting cells. CD4 helper T cells come in, starts activating T and B cells. B cells are made and neutralizing antibodies are made, right? But at the same time, cytotoxic T cells, so CD8 cells, they are also interacting with these APCs. And they're learning how to, what are infected cells and what are not, how to eliminate those infected cells. And this is like many other vaccines, this is where we think more durable immunity may come into play. And then, of course, there are memory T and B cells, which again, we believe will contribute to longer term memory and longer term immunity. So this is how messenger RNA vaccines work. The adenovirus vaccine, so this is your Johnson & Johnson vaccine, or your AstraZeneca vaccine overseas, it's very, very similar. They're still starting with the spike protein gene, but they're actually packaging it instead of a lipid nanoparticle into a human adenovirus, non-replicating adenovirus, and it's in the form of DNA. And so what happens is that adenovirus gets into the cell, the DNA gets into the nucleus, gets turned into messenger RNA, and then it's the same process as I just showed you for the mRNA vaccines. It just has a different carrier, and it starts as DNA. But again, all these immune responses are directed against the spike protein because we think that's the business end of the virus. Okay, so COVID safety. So I'm going to go actually back to the original studies because I think that there is a lot of great real world data out there. And there's more every single day, but there are some downsides of real world data that when you do randomized, placebo-controlled, blinded, double-blinded studies, you can control for some of that bias. But the first thing I wanted to show you was just, again, so you can understand, there are so many different assessments that are done during the course of a trial I just wanted to kind of put the pieces together for you because we're bombarded by with the press every night. We're hearing about another meeting. So once you decide that you're going to do a clinical trial, so once these Moderna or Pfizer and BioNTech or J&J said we're going to do these trials, they provide the FDA a protocol saying this is how we're going to test it, this is whom we're going to test it in, this is our safety plan. And what's called an IND, an investigational new drug application, which basically says, this is our vaccine, this is how we make it, and here are the quality control and other components of manufacturing. If the FDA approves that, then a local institutional review board will look at the bioethical considerations of the trial that's being proposed. And if it gets approval, then it can do the trial. And as the data is being generated, and for the Pfizer trial, it was 7 countries and over 150 sites, and all these sites are simultaneously putting data in, and the data statisticians and the folks are crunching all this data. You have data safety monitoring boards, you have data monitoring committees. Often these are unblinded folks. They know who got vaccine and who did not, and they are on a very frequent basis constantly looking at the safety data and saying either you need to stop the trial for one reason or another, either for futility or because there's a safety signal or because there's such a drastic benefit of the vaccine. and they are constantly looking at data and giving their opinions back to the company and to the regulators. But once the data is, the trial meets a milestone of some sort, they package up the data, they bring it to the FDA reviews it, and then they convene their what's called the VRBPAC, and this is the advisory committee that meets in a public forum. These are objective, outside of the company, outside of the government folks, usually academics, and they have customer representatives and legal representatives and ethics and these sorts of things, ethical representatives. And they are asked to respond to questions from the FDA, which might be something like, does the data support that this vaccine is safe in 5 to 11-year-olds, for example? That might be one question. Another question might be, does this vaccine, is the risk benefit of this vaccine in favor of benefit for 5 to 11-year-olds, something like that. And if they answer in the affirmative and the FDA answers in the affirmative, then it goes to the CDC and the ACIP, which we're all familiar with, and then they vote on how they think the vaccine should be used. So the FDA and the VRBPAC say, is it safe? Is it efficacious? ACIP says, based on that, this is how we would use it. And then if all the thumbs are up, this is when deployment and marketing and use actually occurs. And then there's a whole range of post-marketing commitments or requirements. So there's lots and lots of external groups that are watching over this process. And for COVID in particular, I mean, we've had the VAERS system for a long time, the Vaccine Adverse Event Reporting System for a long time, but now we have numerous other systems that are in place. And listen, I'll be the first to say they are not perfect, but they're good. They can at least collect data that can be in real time analyzed, and signals if they are occurring when thousands and thousands of people are being vaccinated. They can be picked out by the regulators. The regulators can hit pause and they can do an investigation. Very important for COVID-19 is the Be Safe COVID-19 Pregnancy Registry. And by this point, probably 200,000 pregnant women have probably already registered. Last time I looked, there's about 160,000. So there's all sorts of monitoring, safety monitoring that's going on. Okay, so going back to the original trial. So this is the Pfizer vaccine on top, Moderna vaccine on the bottom. They each enrolled over 40,000 people, placebo-controlled, randomized, double-blinded studies. And the data, and they're both messenger RNA vaccines. The Pfizer vaccine is 30 micrograms. The Moderna vaccine is more than 3 times that. It's 100 micrograms. They both use lipid nanoparticles. There's some differences in the lipid nanoparticle, but they're very similar vaccines, and they had very similar side effect profiles. So what we saw is that the predominant local adverse event was pain, and it was typically graded by the people who experienced it as mild to moderate. It started soon after vaccination, and it went away soon after vaccination in most people. Younger people seem to have more local adverse events compared to the older people. In terms of systemic symptoms, both vaccines caused fatigue, headache, and muscle aches. Again, it occurred most often after dose 2, greater than dose 1, and most people said it was mild to moderate. So again, started within 1 to 2 days of vaccination, usually went away within 24 to 48 hours. There are outliers to that, and all of us have our stories, but that's the data from the clinical trials. The Moderna vaccine did have increased reports of chills and joint pain compared to the Pfizer vaccine. The J&J vaccine, which is an adenovirus vector vaccine, very similar. So pain was the predominant local symptom, and again, mild to moderate in nature. But the occurrence of grade 3 events, so the occurrence of severe events, very, very low. That's the gold numbers that are listed above these reactions. And in terms of systemic complaints, very similar: fatigue, headache, muscle aches, again, with very low rates of severe systemic events compared to placebo. Then when we did an age de-escalation, right? So we started in 18 and above, and then we went down to adolescents. Both of the messenger RNA vaccines went down to adolescents. Very, very similar trends in the adolescents as compared to the adults, with pain being the predominant local symptom, fatigue, headache, and muscle aches being the predominant systemic symptoms. All of these, again, worse after dose 2 compared to dose 1. And the vast majority of the time graded as mild to moderate. Moderate pain is defined as basically being, or moderate symptom is, I know I have the symptom, it's interfering with what I have to do, but it's not preventing what I have to do. If it prevents what you have to do, that's considered a severe symptom. And once again, the Moderna vaccine, chills and joint pain were seen in the adolescents with Moderna more than Pfizer. So now this is, now the 5 to 11-year-olds. So this is not in print yet. So what I did was I pulled these from the FDA meeting. And basically what they did was they did 2 studies. They had a cohort 1 and a cohort 2, about 1,500 people in each of those cohorts received the vaccine. First thing to note, it's 1/3 the dose. So this is Pfizer, right? This is the only data we have for any vaccines in this age group at this point. So it's 1/3 the dose. So it's 10 micrograms. There was a placebo group, about 750 people. And basically at the time that they had their data cutoffs, either for cohort 1 or cohort 2, in cohort 1, you had more than 2 months of safety data. In cohort 2, the majority of people had more than 2 weeks of safety data. They put it all together to come up with a safety cohort. And what you see here is, when you look at any pain at the injection site, this is vaccine compared to placebo. So after dose 1, vaccine compared to placebo after dose 2. And so what you see is about 7 people out of 10 report having pain after getting vaccinated. 3 out of 10 people report having pain after getting placebo. Redness occurred with much less frequency, but certainly more redness with vaccine compared to placebo. And the same was true for swelling. About 1 out of 10 people, or 1.5 out of 10 people, had swelling and compared to placebo. But again, a very low rate of severe reactions. Severe reactions are in the red. So these are local symptoms in the 5 to 11-year-old group. These are the systemic reactions in the 5 to 11-year-old group. And again, fatigue and headache were the primary complaints that people had. Muscle aches were a less common complaint, along with some diarrhea, chills, vomiting, etc. Fever occurred in about 6.5% after dose 2, which again, just like in the older folks, was more than what we saw after dose 1, which was about 2.5%. And you can see what we call the severe, so anything greater than 38.9, that occurred with very low frequency, as well as severe other systemic effects. So this is from the FDA. So their conclusion was, local and systemic reactions more common after dose 2 than after dose 1. Pain, fatigue, and headache were the most common. They were mostly graded as mild to moderate by the volunteers, and they lasted 1 to 2 days. In terms of unsolicited, so meaning symptoms that we didn't specifically ask people about, lymphadenopathy, which has also been seen in adults. Lymphadenopathy was the most common, and it was transient and went away. There were some reactions that were graded as hypersensitivity, but they were rash and dermatitis. There was no anaphylaxis in this group. There were no serious adverse events. So these are the type of events that people have to go to the hospital. They have to be admitted to the hospital for death. There were no deaths, this sort of thing, related to vaccination. And there was no myocarditis or pericarditis in this group. I put it in red here, and I'm not going to dig into it, but that is a big ongoing discussion right now. It is one of the reasons why Moderna has not had the FDA give a decision yet on their vaccine in the younger populations, is because of this myocarditis and pericarditis issue, which seems to be, it does seem to be associated with messenger RNA vaccines, and it seems to occur in younger males. So, 18 to 29, 18 to 30 years of age. Okay, so now I want to talk about efficacy and effectiveness. Again, what we're trying to do with vaccines is address a public health burden, which in my view is human suffering and breaking the healthcare system. And so I'm focused on, I think that our vaccines are meant to prevent these moderate to severe symptoms and the outcomes of those symptoms. And I kind of look at it as a pre-Delta and a post-Delta. So this is the pre-Delta. So this is the messenger RNA vaccines. And basically what you see is whether you're looking at 7 days after dose 2 or 14 days after dose 2 in the case of Moderna, very high efficacy. So these Kaplan-Meier curves, blue represents placebo, gray placebo over here, red represents Pfizer's vaccine, blue represents Moderna's vaccine. And it's really good news. They split very early, even after one dose, after about 10 or 11 days. And they diverge very quickly, and they're not coming back together, and very high efficacy. Now, the Johnson & Johnson vaccine was a little different because they were, these trials looked at any COVID of any severity. So if you had a runny nose and your investigator said, swab, do your swab, and the swab came up positive, that's a COVID case. Over here, J&J was looking specifically at moderate to severe COVID. So their overall vaccine efficacy was 66%. And if you looked at severe or critical COVID, it was 85%, which, at the time, one dose vaccine still performing much better than influenza vaccines. Kaplan-Meier curves look good. And it had a very good safety profile. So it still had a place in our armamentarium, and a lot of people early on decided to take it. When we started looking at immunogenicity and efficacy in adolescents, again, these are both of the messenger RNA vaccines. The take home is the immune responses in the 12 to 15-year-olds was as good, if not better, than in the 16 to 25-year-olds. And when you looked at the occurrence of COVID in these trials, very few vaccinated children were getting COVID versus those that got placebo. And then when we looked at the Pfizer vaccine and looked at data out to 6 months, so to get an emergency use authorization, you need at least 2 months of data. To get a full license, you need at least 6 months of data. When you look at the 6 months of data, then it started to get interesting, because what we saw is that although safety was being preserved long-term, vaccine efficacy against any COVID of any severity was declining by about 6% every 2 months, not knowing yet where the leveling off period, leveling off efficacy was going to be. But what was interesting was vaccine efficacy against severe disease was being maintained, and it was being maintained at very high levels in this particular study at 97%. So declining efficacy at preventing infection, declining efficacy at preventing mild disease, but maintained efficacy at preventing severe disease. Now, when you look at the 5 to 11-year-olds, again, it's the same kind of thing. So we're looking at immune responses in 5 to 11-year-olds compared to 16 to 25-year-olds. And what you can see here is the titers of antibodies are very, very similar. So that's good, right? Non-inferior. And also of interest is when you look at a smaller subgroup, so this was 34 kids that were pulled out for a sub-analysis, they looked not only at antibodies 1 month after the second dose to the strain that the vaccine is made from, so the Washington strain, but they also looked at neutralizing antibodies, the ability of the kids' sera to neutralize Delta, and the numbers were very good, and this was very encouraging. And when you looked at the cases that occurred, COVID cases that occurred in the vaccine group compared to the COVID cases that occurred in placebo, small number of cases, 3 in the vaccine group, 16 in placebo, but it still gave you an efficacy of about 91% with a pretty tight confidence interval. And these were the data that allowed the FACP, the FDA, and the CDC to give the thumbs up for 5 to 11-year-olds. Now, what's happened after Delta, because people talk about breakthrough infections. Well, so this is looking at the number of cases, the rates of cases on top, the rates of death on bottom. The black line represents unvaccinated people, and the color, the line, the orange, the blue, and the green lines represent vaccinated people with different the different vaccines that are available in the United States. What you see is Delta entered the country in July, huge uptick both in cases and deaths. You do see breakthroughs, right? So this line, which was flat, almost at 0 for a long time, did increase, but still a substantial difference between, in both cases and deaths, between the unvaccinated and the vaccinated. This looks at hospitalizations in people greater than 18 years of age from January through August, right? So again, here's where we see Delta. Huge uptick in the rate of hospitalization in unvaccinated persons, and a small increase in the rate in fully vaccinated people. If you look at those same trends, cases and deaths, and you look at it by age group, the dotted lines represent unvaccinated people. The solid lines represent fully vaccinated people. You know, what you see here is, again, there is a difference. Vaccinated people have less frequency of being diagnosed with COVID, and they're less often to die from COVID. But you know, as you get in the older age ranges, so here, you know, blue, for example. So this is the 30 to 49-year-old age group. You know, they are going to fare better if they get infected, regardless of whether they're vaccinated or not. But again, there still is a difference. You know, this makes up some of the rationale for why people, even in younger age groups who may be at low risk, should get vaccinated. So how are we doing? So you can see here, as of this morning, the U.S. total, about 67% of the country has gotten at least a full shot, one shot, and we've had fully vaccinated just over 55%, 58%. And in New York, in particular, I pulled that out. It's about 74% of one shot and almost 70% for people being fully vaccinated. Again, this is a heat map. The darker the color means the greater the percentage of vaccination. And you can see that you know, the Northeast, some parts of the Mid-Atlantic and the Midwest are doing pretty good. But I am concerned because people are very optimistic about the 5 to 11-year-old age group. I look at what vaccine uptake has been in the 12 to 17-year-old age group. And if you look at it, it's about 42% of people of adolescents have received at least one dose of vaccine, but only about 32%. And this was in July. They have not published the most recent data. It could be higher. But it's still not where we want it to be. And there clearly seems to be that people are more comfortable the older the child is of getting them vaccinated, which then leads me to wonder what is going to happen now that 5 to 11-year-olds can be vaccinated. And if you look at the Kaiser Family Foundation, which does these great surveys of people, we're kind of split into thirds. We're saying a third of us are saying we're going to get our kids vaccinated immediately. A third of us are saying, we're going to wait and see. You know, as more and more kids get vaccinated, we'll see if the safety is maintained. And then a third of people are basically saying, hell no, I'm not going to do it, only if you make me. So this is where we are. So I'm cautiously optimistic, but I'm also somewhat concerned. So can it get any worse? Well, it could, because we're now entering influenza season. And so is influenza co-infection a thing? It absolutely is a thing. There are tons and tons of case reports and case series. So it is absolutely possible that people can be co-infected with flu and COVID. Does co-infection cause more severe disease? I don't know. Some of the studies you read, yes. Some of the studies you read, no. So I think that story still needs to be finished. What I will show you is, you know, this was the 2019-2020 influenza season. And again, I picked 1 week, the first week of the calendar year. It's a heat map. So blue and maroon and red, that's very high influenza activity. So that's what it was in 2019-2020. This is what it was in the 2020-2021 season. And we can all think about why that probably happened, which was because, yeah, well, people weren't getting together. People were wearing masks. People were washing their hands. No one was going to school. People were teleworking. No social venues were open. Well, most of those things have all now changed. So, you know, this is where we are in the current influenza season. Oh, I didn't change this. It's supposed to be 2021-22. Sorry about that. This is where we were last year. But this is, you know, these right here represent what the years were leading up to this. So, you know, we'll see with the removal, for the most part, of those non-pharmaceutical interventions. We'll see what vaccine uptake looks like, and we'll see what you know, what people do and how people behave. But how are we going to manage this flu and COVID vaccine thing? Well, first of all, the FDA, the VRBPAC, has selected the strains. We're going to have mostly quadrivalent vaccines available, both egg-based and cell or recombinant-based vaccines in the U.S. They're very similar, except for one component of the H1N1 vaccine. fortunately, there is data. So we've heard that, and the CDC, getting right to the punchline, you can safely co-administer flu and COVID vaccines at the same time. But there is data. So there's data with the Novavax vaccine, which is not available in the US, but the conclusion of this study with a couple hundred people, no safety concerns, no reduction in influenza immunogenicity, a mild reduction in their COVID vaccine immunogenicity, but in the context of this study, COVID vaccine efficacy seemed to be preserved. So this was a subgroup within a larger COVID vaccine trial. And then this was a study that it's a preprint from the Lancet looking at the chimp adenovirus. So this is the Oxford AstraZeneca vaccine and the BNT vaccine. So this is the Pfizer BioNTech vaccine. And they did a phase 4 randomized controlled trial, which was also blinded, looking at the impact of co-administration. And there were no safety concerns, no reduction in influenza immunogenicity, and no reduction in COVID vaccine immunogenicity. So I think there's data to support that we can safely co-administer vaccines. So in conclusion, SARS-CoV, it's the latest emerging coronavirus causing worldwide human suffering. So SARS-CoV-1, you all remember, and Middle Eastern Respiratory Syndrome coronavirus, so MERS-CoV. So this is the third. There's lots of vaccine development activity going on out there, as I showed you, and a number receiving, one with a full approval and multiple receiving authorization. Even with Delta, these vaccines work to prevent what I think is the public health burden, which is severe disease, hospitalization, and death. It'll be interesting to see how vaccinating 5 to 11-year-olds could potentially impact the epidemic. I still don't know if we've conclusively answered what role young children play in transmission. And then, the bottom line, flu and COVID vaccine co-administration is okay. And it's okay not just because people say it's okay because there's actually some data. So I will thank you very much for your time and your attention. And I hope I left a little bit of time for questions if you have them. So back to you, Amanda.
Dr. Thomas, that was absolutely superb. Thank you very much. It was very informative. And I'm going to let Amanda and Lisa man the chat lines, but I'm going to take the prerogative as host to ask the first question. And this is a question I get asked by my staff and my participants all the time. And that is, will the COVID vaccination be a yearly event from now on?
The answer, which will be unsatisfying, is I don't know. This is the reason why these trials are multiple years, though. We need to, and now that a booster dose has been introduced into the trial, that certainly impacts, I think, the length of follow-up that we're going to need to understand whether or not this is going to be annual, whether or not this is going to be every few years. It's also going to be dependent a little bit on what does the mutation landscape look like, right? So I think it is good news that we are still able to kind of cross protect, if you will, using a different strain. And we're still able to cross protect against Delta, but vaccines mutate. That's what they do, and there is the potential that a mutation could come along which can efficiently escape vaccine-induced immunity. So, my guess is that if you just had a 2-dose vaccine, you probably definitely would be getting a booster at some point in your future. If it's a 3-dose vaccine, or if it's a 2-dose in a 6-month, 6 to 12-month booster, I'm not sure what the immune tail looks like. So unfortunately, I can't answer definitively.
All right, thank you. Amanda, do you have some questions lined up?
And this is Lisa. Let me just, I'll read what's in chat. One of the things a person who is skeptical of the danger from COVID-19 said to me the other day was that the degree to which the spike protein can mutate will soon be exhausted. The person did not have a response to that. Dr. Thomas, do you have a response to that?
That the ability of the virus to mutate will at some point hit a wall and it won't be able to mutate anymore?
Correct.
Yeah, well, I mean, that would be nice, but there's, you know, hundreds of years of virology that go against that. And, you know, I just think you can just look at our it's a different type of virus, but you can look at our influenza experience to see what all the possibilities are for mutations in different parts of the virus. And it's not just about the spike protein. There are mutations occurring in other parts of the viral genome, which have significant implications. And because, you know, there are parts of the virus not in the spike protein that alter our immune system, which make us a good host for the virus to infect. And that's also, they're also mutating. So unfortunately, I cannot share that optimism.
Okay, the next question is, what part of the body was affected with the lymphadenopathy?
The lymphadenopathy? Yeah, so the axilla. So it was basically the armpit of the side that you got vaccinated on. That was the main issue. And it was an actual, I mean, it's a real issue and a practical issue because, you know, just one example, women who were going for breast cancer screening who had just been vaccinated showing up with lymphadenopathy, that's not, or any kind of screening for any kind of malignancies, that's problematic. And so I know in the hem-onc community, there were some recommendations to avoid doing those types of surveillance scans or mammograms, et cetera, around the time of vaccination.
Okay, let's see. Are there reports of delayed onset of adverse consequences greater than the 2 weeks after administration?
Yeah, so it's interesting. If you look at all the vaccines that have been licensed, certainly in the United States, the vast majority of any adverse event that is realized from a vaccine occurs within 6 weeks of administration, which is why the FDA said, for you to get an emergency use authorization, you have to show us at least 8 weeks of data, because the majority of adverse events that we see are within that 6 week period. And so that being said, though, this is another reason why the vaccine developers are required to do these longer studies to collect safety data for a longer period of time. And they are probably, I'm not part of these conversations or negotiations, but it's probably why they are going to have pharmacovigilance requirements and post-marketing requirements and post-marketing commitments to do long-term safety analyses.
Thank you. Is it better for the patient not to mix vaccines? What combination are acceptable and in which order?
Yeah, the mixing and matching, or what we call heterologous prime boost. So my recommendation is that people should stay with the same manufacturer if they can. So if you got J&J, or if you got Pfizer, get Pfizer. If you got Moderna, try to get Moderna. And I only say that because the majority of the data is around, you know, priming and boosting with the same manufacturer. So I like to follow the data. That's where the majority of the data is. If for some reason that is not a possibility for you, then the next thing I would do is try to stay within the same class. So stay within messenger RNA, or you can stay within the adenovirus vector if there's no other contraindication of getting an adenovirus vector boost. So Moderna, Pfizer, Moderna, if you can. Of note, the Pfizer boost is the same dose, 30 micrograms. The Moderna boost that was tested is 50 micrograms. It's half the original dose. So that's what I do. And then the third is, if neither of those are possible and you're a person at risk of infection, or you're a person at risk of a bad outcome if you get infected, then you can, I think it's okay to mix and match and get a different vaccine than you got for your first dose. I wouldn't, in terms of order, I wouldn't kind of prospectively make the decision to mix and match. So regardless, so the order will be whatever you were able to get as your first dose. And, you know, and fortunately there is data. There's data in Europe. There's data from our NIH that support both the safety and the immunogenicity of mixing and matching. And it's not, just so you know, it's not a new phenomenon. We look at heterologous prime boost strategies with lots of different vaccines because there's immunologic benefits behind it potentially.
Thank you. Are there reports of delayed onset of adverse consequences greater than 2 weeks after administration?
Yeah, I think we answered that one.
Did we? Okay, thank you. I'm sorry.
That's right.
Let's see. For mammogram screening, is there a specific time recommendation?
Yeah, so I would talk to your primary care doctor. I would talk to your oncologist, but I believe it is at least 4 weeks. It certainly is until the lymph. If you develop, I think it's at least 4 weeks from vaccination if nothing occurs. If you do get lymphadenopathy, for example, or you have some kind of symptomatology, adverse reactions after vaccination, they should be completely resolved before you get evaluated. But I believe it's at least 4 weeks.
Okay. How about for patients who received J&J as first dose, recommend mRNA as booster versus a second dose of JJ?
Yeah, so the Johnson & Johnson, as we know, I mean, there was a lot of uptake, and then there were these concerns about thromboses. There were concerns about the syndrome of thromboses with thrombocytopenia. Those and it was in a certain age demographic and population, young women. So I think which is why before I said, if there's no contraindication to getting a booster with J&J, you could certainly do that. There are studies that, there's a study that shows it's safe. There are some people, I will say, there are some scientists and vaccine developers who think a booster with messenger RNA is optimal. I just don't feel like I have enough data to make that conclusion. So I would reflex to where is the most data, and the most data is with a J&J followed by J&J if there is not some contraindication or reason why you couldn't get J&J.
Thank you. Bear with me. I'm sorry. For mammogram screening, is there a specific time recommendation?
That one I think you got to.
Did I? Okay,
Yep.
Thank you. I'm sorry. There's quite a few here. I have a melanoma patient on treatment that had Janssen. Should I have him get Pfizer rather than repeat Janssen because he is immunocompromised?
Yeah, so I don't. So there are. I don't know. I wouldn't call them phase 4, and I wouldn't call them post-marketing. I would say there are real-world studies in all sorts of different populations of people, to include patients that have different types of malignancies. It is, and this is a tough one because we did not enroll. We enrolled people who had active and past history of cancer, but we did not enroll people who were immunosuppressed either from medications they were taking or from the disease itself. So if you have some, which is kind of unfortunate, right, in a way, because they're the people that really need to be immunized. So what I have seen in I'm not an oncologist, but what I've seen in the our hemonc community is that it's a very individualized approach. And if they are immunosuppressed because of a treatment regimen, they try to avoid vaccinating during that treatment regimen. Or if the treatment regimen for the underlying malignancy can wait or be pushed, then they'll vaccinate them first, and then they will proceed with that therapy. The other thing is we know, people with solid tumors seem to respond well to vaccination. People with Hematologic malignancies, though, they do not seem to respond as well, which is why, back in August, the government allowed a third dose in that group. So it really is an individualized discussion, and it depends upon why the patient is immunosuppressed, whether it's from treatment or whether it's underlying disease. I would say if the person has thrombocytopenia, I probably would not use J&J. I would give them a messenger RNA vaccine.
Thank you so much, Dr. Thomas. This is Amanda back again, and we've got a lot of great questions and a lot of great information. We just wanted to end. We are over time, so we will finish with one that had come in that asks, for people who have had COVID in the past and are reluctant to get the vaccine, given that we still do not have an immune correlate, what data can we provide to help guide the patient, given that breakthrough infections are being seen with all 3 COVID vaccines?
So breakthrough infections are occurring, but breakthrough infections that result in hospitalization, severe disease, or death are occurring with a much less frequency than in unvaccinated people. And they are occurring in the breakthrough infections that result in those outcomes are occurring in people that unfortunately you would expect, people that are older, people that have underlying comorbidities. So I think we have to, when we say breakthrough infections, we have to put a finer point on what we're on what we're talking about. The other thing I'd say is that there are cohorts that have followed, and Upstate has one of them, that have followed COVID survivors, and what we see is that a significant percentage of them, 30 to 40% by 6 months, have definitely decreased antibody titers. Some have undetectable antibody titers. Some of them have decrements in the quality of the antibodies that they have, because not all infections are created equal, in terms of how you were infected and the dose you got, and these sorts of things. And so people have a variable immune responses, and the durability is in question. And there's actually, and so that's one thing. The other thing is we had people, so everyone was tested before they got vaccinated. And so we had a group of people who were COVID positive, either by antibody or PCR at baseline, that got randomized to the placebo group. Nobody knew who they were. And so we have efficacy of previous infection against reinfection, and it's less, and it's lower than what we get from vaccination. I mean, if you get natural infection and then get vaccinated, you are probably the most immune person in the room compared to people who just get vaccinated or just get natural infection. I know there's some data out there that speaks to, natural infection is better than vaccination, but I think that's going to be a wait and see till there's more studies, because I do not believe that that's going to be the conclusion of that story.